In this video clip, I'm going to talk a little bit about techniques that are used in the laboratory for culturing and identifying viruses, like the ones that would cause infections in humans. So things work a little bit differently with viruses than they do with organisms like bacteria and fungi that you can grow in laboratories on agar petri dishes. You can't do that with viruses because viruses need a host in order to survive. And if you're interested in culturing viruses that cause infections in humans, you're going to need petri dishes of host cells that are appropriate for those viruses. So human cells you can't just culture in a laboratory quite as easily as you can little microorganisms like bacteria and fungi. And typically what you will do in a laboratory is if you want to culture viruses, you will grow sheets of cells, and very often they're human tumor cell lines, believe it or not. Many of those are good hosts for viruses that cause infections in humans, and you can grow those in little, like what you're seeing here in this picture, that's a little plastic flask, and that red fluid that you're seeing there is a culture medium that is appropriate for growing these cell lines. And the problem there is that culture medium tends to be very expensive, so it's a lot more expensive to grow human cells in a laboratory than something like bacteria, which grow like gangbusters, and you can grow them relatively cheaply. Also, human cells, if you're growing them in a laboratory, they don't divide nearly as quickly as something like bacteria or yeast would, and that's another thing that makes this more difficult and more expensive, because even bacteria, so we talked about back in chapter 3 how bacteria, when they're happy, they're getting plenty of nutrients, they're in a nice warm incubating environment. One cell can divide into two, in some cases maybe every 20 or 30 minutes might be the generation time. When you're thinking about a human cell line that you're cultivating in the laboratory as a host for viruses, the generation time may be more like 24 hours or even a little bit longer. So it's more difficult to expand out a culture of these human host cells that would be useful. And then you've got to be a very highly skilled laboratory worker to grow these types of cells. These cultures get contaminated really easily. You've got to have much more expensive specialized equipment to do this. So not quite as routine. You won't walk into a typical clinical microbiology laboratory in a medical setting and see a lot of virus culture going on. All right, so this is a picture of some uninfected cells that you would grow poliovirus in. So poliovirus, of course, can cause paralysis, and we have good vaccines for it, and it's largely been eliminated off of the planet. But you can grow poliovirus in the laboratory in a number of different cell types, human cell lines, and then some other primate cell lines. And here you see a sheet of host cells for poliovirus, and so right there, and right there, right there, right there, each of those things that kind of looks like a tile is an individual cell. The darker purple spot in the center is the nucleus. And then if you had a culture like this of those cells in the laboratory and you put some polioviruses in there, came back 12, 18, 24 hours later, this is probably what you would see if you put enough of those viruses in there. The cells round up, they start to detach from the surface on which they're growing, and they look very sick and unhappy. So these are cytopathic effects, like I mentioned in the last video clip. Poliovirus causes very severe cytopathic effects on cells. All right, so now more typically, when viruses get diagnosed these days in a clinical laboratory type setting, they're not often cultured. Instead, we're using other techniques. So if you guys remember the polymerase chain reaction, PCR technique, that we talked a little bit about back in chapter 8, that is a very specific DNA-based technique that can, in a little tiny test tube, amplify very specific sections of DNA. And so basically there are now very sophisticated assays that can be done. You can take a sample from a patient, You design PCR primers that home in on some gene or some section of a specific virus's genome. And if those viral genomes are present in the sample, the PCR will amplify them so that they can be detected, using a technique like agarose gel electrophoresis, which you guys are doing as part of your Learn Smart lab activities for chapters 8 and 5. And you might be wondering about those RNA viruses, because we said PCR amplifies DNA, but interestingly, that good old enzyme we learned about, reverse transcriptase, that HIV and its relatives use as part of their life cycles, that can be used artificially to convert. If you have an RNA virus, its RNA genome can be converted over to DNA using that enzyme, and then you just run the standard PCR on it, so you can actually amplify RNA virus genomes as well. All right, so are those types of techniques being routinely done in clinical laboratory settings? Not yet, but probably not too far down the road. We will see that happening. There are companies, though, that's how they make their livelihood, is by developing and carrying out very sophisticated PCR assays that can detect viruses and other types of pathogens as well. And a company called Diatherix here in Huntsville over at the Hudson Alpha Institute of Biotechnology, that's its claim to fame. That's one of the things that it does, is very sophisticated, sensitive PCR assays for various patient problems, including viral infections. And if you think about it, that's very different from what we're used to. Usually if you go to the doctor and your doctor thinks you have a viral infection, unless it's one of a handful of viruses that we have good rapid tests for, you're probably not going to know exactly what kind of virus you were infected with. But down the road, as we have more of these types of techniques available, you will know more and more exactly what kind of virus you're infected with, and that'll be important as well as we develop more and more treatments for viral infections. We're going to need to know exactly what we're infected with so we can get the correct medication. All right, so then there are assays called immunodiagnostic assays. We've mentioned this a little bit before in previous video lectures. So these actually make use of antibodies. Antibodies are protein weapons that the immune systems of animals generate to help fight infections, and they attach to very, very, very specific molecules, such as the molecules that would be on the surface of a virus. And once they do that, you go through typically like a color change type reaction as part of the assay, and that will give you a yes or no answer on whether a patient is infected with a particular virus. So if you, like what happened to me last winter, my doctor thought I might have the flu, so they stuck that awful cotton swab up my nose and took a sample and took it off to the lab, and about 20 minutes later they were able to come back and tell me, no, you didn't test positive for the flu. So if you've had that done to you anytime recently, that was a type of immunodiagnostic assay. It's using antibodies that attach to the flu viruses, and that triggers a color change reaction and gives your physician a yes or no answer as far as whether you're infected with the influenza virus. And they have tests like that for HIV as well. Maybe you've had a rapid strep test. I had one of those at the same time I had the flu test, so I had to get gagged with the swab in the back of the throat as well. Home pregnancy tests are a type of immunodiagnostic assay. Blood typing is a type of immunodiagnostic assay. So these have been used a lot. They're generally pretty quick and they're very sensitive, and they're a lot more widely used in typical clinical laboratories. But we don't have these types of assays available for every type of virus that you might be infected with. They're available for some of the more common ones out there. Oh, I'll mention this as well. And some immunodiagnostic assays are not looking for the virus. They're trying to detect the virus from a sample that has come from your body. Some of them actually look to see whether you have made antibodies against that virus, because if you have been exposed to the virus, then your immune system will have responded, and you will have manufactured your own antibody weapons to try to combat that viral infection. And so there are some assays that actually kind of give indirect evidence of a viral infection by looking for the antibody weapons that your own immune system made against it. Those types of assays are useful when virus levels tend to be very, very low in a patient's body fluids. All right, so I'm going to go ahead and wrap up that video lecture here. The next video lecture for Chapter 5, we're going to talk about other non-cellular infectious agents, and we're mainly going to be focusing in on prions, which are kind of strange, but very interesting. So that'll be the topic for our next video lecture.